All right, so today I'm going to do a quick run through of photosynthesis. We're going to talk about a little more of the in-depth parts. We're not going to get too in-depth, but we're going to talk a little bit more about where our equation comes from and how the different parts of photosynthesis kind of interact with one another within the chloroplast. So we have talked about both photosynthesis and cellular respiration. We talked about how the goal of the chloroplast is to turn solar energy into chemical energy in the form of glucose. It's also going to produce oxygen as a byproduct, but it's going to take some other bits and pieces to kind of make that come together. It's not just an instant process. It takes a little bit. So the main discussion that we're going to have is kind of what goes in and what comes out of photosynthesis. So our main equation for photosynthesis is going to be this 6 carbon dioxide plus 6 carbon dioxide plus 6 water in the presence of sunlight gives us oxygen, O2, and 6 molecules of C6H12O6, this being our glucose molecule. Glucose, again, is a carbohydrate. We saw this couple units back, but glucose is going to be our sugar in this case that is produced from photosynthesis. So, if nothing else, please try to remember this equation. This one's going to be very important. We're also going to revisit it when we come through cellular respiration. We're going to see that this kind of flips and uses oxygen and and glucose to make carbon dioxide and water. So this is going to happen, like I said, in the chloroplast, and there's a couple parts that we're going to talk about, in particular the thylakoid and the stroma. The thylakoids are going to look like these Mentos. They're just kind of stacked on top of one another. Someone else in one of my blocks said that they kind of looked like pancakes stacked on one another. But they are going to be these little membrane discs that are found within the chloroplast. The other part we're going to talk about then is the stroma. The stroma is going to be kind of the cellular or the interchloroplast kind of gel. It's kind of like the cytoplasm was in our plant cells. If you remember looking at your aquatic plant leaves, you saw a whole lot of chloroplast moving around in there. We are going to be focusing on this, but the stroma is going to be the cytoplasm-esque version for the chloroplast. There are going to be two different, actually we're talking about chlorophyll. Photosynthesis, okay, there we go. So photosynthesis is going to occur in two different stages, the light reactions or the light dependent reactions and the Calvin cycle. The Calvin cycle is occasionally called the light independent reactions or the dark reactions. I don't typically call it the dark reaction, but light independent is another good name for it. In the light dependent reactions, it's going to occur in the thylakoid, those little discs. What we're going to do is we are going to use a whole bunch of different systems called photosystems, and we are going to extract the sun's light. We're going to take this light and absorb it into our photosystems to energize some electrons. This is going to happen in our chlorophyll, which I won't get too much into, but chlorophyll is the pigment that makes plants green. It's also going to be the energy absorber for the plant cell. So when the photosystems get hit by energy, or hit by sunlight, they're going to excite a bunch of electrons. And this is going to do a couple different things. Number one, this excited electron is going to break water in half. We said a couple of units ago that water is H2O. So we are going to produce hydrogen, H, and we're going to produce oxygen, O. So now you're starting to see a couple of the different parts kind of show up. So the oxygen is going to be removed. It's going to be spit out. It's kind of a waste product. The hydrogens, on the other hand, we're going to hold on to. They're just going to kind of collect on this lumen in this inner area of the thylakoid. Then what you're going to do is move the electrons from the photosystem through a whole bunch of things to another photosystem. When this happens, it's going to shuttle some more hydrogen from the outside of the outside of the thylakoid into the thylakoid. So we're seeing that we're getting a lot of hydrogen buildup on the inside here, and that's going to play an important role we'll see later. When the electrons go from photosystem 1 all the way over to photosystem 2 all the way over to photosystem 1, it's a little backwards, this is going to get hit by sunlight again. It's going to excite the electrons, but this time instead of adding more hydrogen, we are going to take these excited electrons, we're going to shove them into something called NADPH. This is going to be our electron shuttle. It's kind of like if you take the bus anywhere, the NADPH is going to be the kind of electron shuttle from the light dependent reactions all the way over to the Calvin cycle. So it's going to charge up. It's going to go from NADP plus to NADPH. And at the same time, you again have all these hydrogens. What you're going to do with them is you're going to give them a way out. Now, if we just give them a random door, it's not really going to do anything. There's no point in building this high amount of hydrogen on one side if we're not going to just let it go normally. So what you do is you add a turnstile to it, or a revolving door. In this case, we're going to be using an ATP synthase. This is a big protein, and it's going to let the hydrogen come out, and as the hydrogen come out, they are going to turn this little wheel on top. As this wheel turns, it creates energy that is going to be stored into our favorite molecule, ADP. This ADP plus the energy from the ATP synthase is going to charge it up like a battery, and we are going to get ATP, our cellular energy. So, just to recap, we are going to excite a bunch of photosystems. They're going to charge a bunch of electrons. Some of those electrons are going to be used to break water into hydrogen and oxygen. The oxygen is going to leave. The electrons then are going to get shuttled and funneled into NADPH. It's going to charge up NADP plus into NADPH. The rest of the hydrogen then is going to come out. It's going to charge our ADP, our adenosine diphosphate, add that last phosphate back on, and charge it into ATP, adenosine triphosphate. So in total, our light-dependent reactions happen in the thylakoid. The reactants are going to be a couple different things. We have sunlight, we have water, we have ADP, we have a molecule that is coming in, it's uncharged, it's going to get charged here. Same thing with NADP plus. These are both going to be kind of the dead battery equivalents to both of these molecules. And once we finish with the light-dependent reactions, we are left with oxygen. We're left with ATP plus an additional phosphate gives us ATP, which is our very important molecule, and NADPH. So take a minute to jot all this down. If nothing else, this is going to be your main review guide for photosynthesis. Like I said, ATP and NADPH are going to be very important products for the light-independent reactions. The whole point of the light-independent reactions is going to be making energy. You can see that we are literally just taking water and sunlight and converting it into some form of energy that we can use further. Now you do see that we make ATP. We said that ATP was mostly made in cellular respiration. Instead, we're making glucose here. So in this case, we need a little bit of energy that we're going to see here in the Calvin cycle to get the Calvin cycle to actually work. In order for us to get glucose, we need a tiny bit of energy. So you'll see we'll make a couple, maybe 2 molecules of ATP in this stage. In cellular respiration, here probably tomorrow, if not the following day, you'll see that ATP in cellular respiration, we're going to make about 36 to 38 molecules of ATP. A whole lot more in cellular respiration, which is why we devote cellular respiration to making ATP instead of just using reactions like this. So we're going to get a whole lot more bang for our buck when we do cellular respiration. Okay. So the next stage then is the Calvin cycle. Now we see the name cycle. So a cycle is something that is going to repeat. It's going to kind of regenerate itself so that it can keep happening over and over and over and over again. So this is going to occur in the stroma. So it's going to happen in the kind of outer layers of the not the thylakoid, but in this kind of gel within the cytoplasm. And in this case, we're going to take carbon dioxide, another reactant for this whole reaction. We're going to combine it with something called RUBP to form 12 molecules of carbon. We're going to make 3 organic molecules. We are then going to take our ATP and NADPH, which we said came from the light reactions. We said they came from over there. We're going to turn these 3 carbon molecules into molecules of something called G3P. Now, the name for this is actually glycol 3-phosphate, I believe. You don't have to remember that. In fact, the G3P is going to be used for something here in a minute that's going to be a whole lot more important than G3P ever will be. But we are going to use this ATP and NADPH, and we are going to charge our molecule to make G3P. So we have 12 molecules. 2 of those are going to be released out of the cycle. Now we have a 3 carbon molecule. 1, 2, 3. And a 3 carbon molecule. We add 3 and 3. We get 6. What molecule is made out of 6 carbon? Give you a second to think about it. C6H12O6, glucose. So 2 of these molecules are going to be spit back out. There we go. 2 of these molecules are going to be spit back out and combined together through a couple more steps to make glucose. The other 10 are going to be cycled back in here, and they are going to regenerate RUBP. So this RUBP is going to be kind of an enzyme that helps this whole reaction happen. So you are going to take more energy to do it, which is all going to come from the light reactions, but we are going to regenerate RUBP, which is going to restart our Calvin cycle. So you can see that as we add more carbon dioxide, we get more G3P. The G3P gets spit out as glucose. The rest of it goes back and restarts the cycle. So it's going to keep happening over and over and over again. The RUBP never goes away. It's just transformed into a different source. Okay. So, looking at our second part, the Calvin cycle, take a couple seconds, pause the video, and fill out the Calvin cycle. Where does it happen? What are the things that go into it, the reactants? What comes out as products? And kind of what is the main job of the Calvin cycle? Make sure that it's still fixed. Okay. So, the Calvin cycle, the main job of the Calvin cycle is going to be for creating glucose. The whole point of the Calvin cycle is just to make glucose. It's going to happen in the stroma, the kind of cytoplasm of the chloroplast. The reactants, the things that are going to go in, are our carbon dioxide, our NADPH, our charged version. So a little lightning bolt there. And our ATP. We also have Rubisco here on the side. Oh, RUBP and Rubisco, but RUBP. But since he gets recycled, we're not going to really count him in this. He's just kind of a facilitator of this whole reaction. The products then, we are going to take our carbon dioxide and we are going to make G3P, which turns into our sugar in the form of glucose. If I can write. Glucose. We are also going to lose the charge in the ATP and the NADPH. So we are going to get ADP, adenosine diphosphate, and we are going to get NADP plus. So as you can see, if I quickly jot everything up here, so we had water, oxygen, so if I just quickly jot up all of our products, you can see the reactants of one, at least the ATP and the NADP plus, are going to be the product of the other. We are going to take this ATP and the NADPH and constantly cycle it between the light-dependent reactions and the Calvin cycle. We are going to make this energy in the light-dependent reactions, shuttle it over to the Calvin cycle, dump the energy, and turn it back to the light-dependent reactions to make more energy. So we are literally funneling the sun's energy into this reaction where the ADP and the NADP plus are going to be the middlemen. They're going to take this solar energy in the form of excited electrons, and they're going to funnel it over to the Calvin cycle where we can pack all of this energy into glucose. So hopefully this shows you kind of where all of this is coming from. We are taking the chemical reaction from the solar energy all the way over to the chemical glucose. So for your quiz, I would study all of this as well as your reaction, our equation. Our carbon dioxide plus water in the presence of sunlight gives us glucose, C6H12O6, plus oxygen, O2. And this is just kind of reiterating. We take the sunlight, we put it into the thylakoid. Again, kind of looks like a stack of pancakes. Add water, break the water in half to get hydrogen and oxygen. Oxygen gets split out. The hydrogen then is going to get filtered through to produce ATP. The energy from the light is going to excite a bunch of electrons to make NADPH. Then we shuttle all of this over to the Calvin cycle where we take RUBP, combine it with our carbon dioxide to make a whole bunch of different molecules. We're not going to go into those molecules into too detail. We're going to make our G3P. Some of that's going to go back and regenerate RUBP, and the rest is going to make our sugar. So, if you have any questions, feel free to do the additional practice that we did today. Please work through all of that. If you have any questions, feel free to ask myself or one of the teachers helping you out. We are more than welcome to help you through this. This is a little complex, so ironing out any details that you may have missed, any misconceptions, things like that, are going to be very helpful for you both on the Keystone and in the short term taking this quiz and eventually this unit test. All right. Thanks for joining me.